Today I'm going to describe for you how creating crosses between two different populations or two different species and examining the genotypes of their hybrids can help us better understand and locate loci that are involved in hybrid genetic incompatibilities. So in this case, what I've diagrammed is a P0 generation between two different populations, population A and population B. And each of these populations of worms, in this case, I've diagrammed a cell, and in this case, 3 different chromosomes, 1, 2, and 3. And I've indicated here that there are 3 different genes, say gene F, gene G, and GH on those 3 different chromosomes, and that there are differences genetically between these 2 populations. One relates to the colors. So the colors of F, the red, indicates a de novo or new mutation in that population. The same is true for the yellow mutation and allele of gene G, and in population B, a new mutation, not the ancestral or wild type mutation, at gene H. And the idea here is that this is a type of Dobzhansky-Muller incompatibility where population A has been in isolation for some time and has accumulated the mutations red and yellow near or in the F and G genes respectively, while population B has accumulated a mutation, the blue mutation, in or near allele H. And in this scenario, the incompatibility exists between the blue and the red alleles. So in these initial populations, with 2 alleles of the red version of gene F, cartooned here as proteins, those are shown to interact with the proteins produced from gene H. And again, H here is the wild type version, the gray or the ancestral version. So these 2 proteins can work functionally together within the context of population A's genotype. And gene G, which produces protein G, has no role in this process. So this is going to be an example of a neutral mutation, the yellow mutation. In population B, it's homozygous for the blue mutation in gene H, which creates a slightly different version or shape of protein H. And so within the context of population B, with the wild type or ancestral version of F shown in gray, and the new derived or blue version of gene H, those proteins can still fit together and interact and conduct cellular business. And again, gene G, which is here is shown as wild type, is irrelevant to the interaction between F and H. So again, the incompatibility is between the blue and the red alleles in this cartoon example, where those 2 proteins, if brought together in the same organism, don't fit together properly. And so that cellular process that those proteins undertake probably won't happen, and that could cause, in this example, hybrid dysfunction. And to be more realistic about how this process actually works in genetic research, I'll point out that in these 2 populations, you don't know at the beginning of the experiment where the genes are that might be incompatible, if there are any genes that are incompatible with each other. So, for example, we wouldn't know in advance that there are these colored alleles of derived mutations compared to the gray versions of the ancestral mutations. So instead of focusing on those for the next little bit of this example, I'm going to remove those color codes and just indicate that in these 2 different populations, there are differences in insertion deletion or indel polymorphisms that are not necessarily causal. That is, these mutations don't necessarily cause hybrid dysfunction, but they are maybe located near genes that do. So instead of tracking the causal genes because we don't know what they are or where they are, instead we can genotype hybrid offspring for these SNPs and indels, in this case indels, that are located all over the genome. So we don't have to know exactly where the incompatible loci are to begin this process, but we do have to either be lucky about which of these mutations that we choose to genotype, or genotype enough mutations throughout the entire genome across all of the chromosomes instead of what I've diagrammed here, which is one locus per chromosome. And as a quick indicator of what an indel is, that stands for insertion deletion. And what this indicates is that there can be regions of a chromosome that have undergone a deletion, in this case, a 100 base pair deletion, which is often indicated with the Greek letter delta for deletion. That's 100 base pairs on chromosome 1 that's missing in population A, but is present in population B. So in this example, we have 3 different indels, one on each chromosome, and those can be used to track the inheritance of which population's chromosomes are transmitted into the offspring. And for completeness, I will point out that there is this 100 base pair deletion or insertion on chromosome 1 in population B. That's why we call them insertions or deletions. We don't know if there's a loss of DNA on this chromosome or a gain of 100 base pairs on the homologous chromosome in an adjacent population. So insertion or deletion. 100 base pairs on chromosome 1, an indel of lack of 200 base pairs on chromosome G or chromosome 2 near gene G in population A, where it's present on chromosome 2 in population B. And then this time in population B, a small insertion or deletion, lack of 50 base pairs of DNA on chromosome 3 in population B, where that DNA is present on chromosome 3 in population A. So we have 3 different indels on 3 different chromosomes, and for this example, they're all different sizes, so it will be easy to track the transmission of these indel polymorphisms or mutations as these 2 mutants, wild type really, but they're mutants, are crossed together to see what happens to their offspring. And when those ancestral genotypes, population A and B, the P0 generation, are mated together, they produce hybrid F1 offspring. So here are 2 genetically identical, essentially, sibling F1 individuals. And if we take a look at their genotypes and their protein compositions, we can see that an individual F1 is completely heterozygous. If you begin with 2 pure breeding or genetically homozygous parents, as was the case here, then the offspring will be heterozygous. They'll have a derived mutation allele on each of the 3 chromosomes we're looking at, and those are linked to or next to or adjacent on the chromosome to those indels. So in this cartoon, although it's not necessarily the case, I've identified that each derived mutation, the colored blocks, is linked to or on the same chromosome from the deletion version of an indel, the smaller version. And we can take a look also at what this genotype means for the phenotype of this organism, shown with the cartooned proteins here. So in this case, because of the way that these proteins evolved, in a Dobzhansky-Muller incompatibility approach, heterozygotes are often fit, they're often healthy, because even though there's a red version of gene F and it interacts poorly with the blue version of gene H, there is still a wild type version of F, this protein, to interact with the derived or mutant allele of H in blue. And the same is true for the red F, which wouldn't interact with this blue H, because in this example, that H has this little sticky up bit caused by the mutation that causes the blue allele. And that would not allow this protein to fit together with the red protein F. So in a heterozygote, in this case, if this interaction between blue H and red F is recessive, where both copies of each allele would have to be red, like red F over red F and blue H over blue H, that would probably be bad. And that's where we're going to see in this example, there is a hybrid dysfunction that occurs. But often in heterozygotes, you won't see that. So often in the F1 generation of a cross, you won't necessarily encounter any evidence of hybrid dysfunction, even if it might exist in future generations. But again, we don't know the colors and the mutations or even the genes in advance of doing these sorts of experiments. So we have to take an unbiased approach at searching the genomes for regions that might contain genetically incompatible alleles. And that's why, again, I've taken away the colors and the proteins to represent that this is really a generic case of taking two pure breeding parents, creating heterozygous offspring, and mating them together to produce the F2 generation. And that's because in the F2 generation is the first time you could get that genotype that I was just mentioning, where you'd have, for example, 2 blue H's and 2 red F's. So it's the F2 generation, the first time that you could get that potentially deleterious interaction between alleles. Now let's zoom in a little bit and look at in greater detail at what's happening at the molecular level in these genotypes. So for the purposes of PCR genotyping, which is a technique of using these DNA primers, shown as the half arrows, to amplify parts of chromosomes to inquire about their length. So in this case, for example, the distance on the chromosome between the start and the end of primers 3F on the left and 3R reverse, forward and reverse, F and R, on the right, that distance in this population is about 450 base pairs long. And if we go look at what happens in the other parent, because there's this mutation, this deletion, compared to population, the first population, here, because there's a deletion of 50 base pairs, that will only produce, using PCR with the exact same primers, excuse me, not 300, but 400 base pair long piece of PCR product. So in other words, the distance between the two primers in this population is 400 base pairs in population B and 450 base pairs in population A. And we can see that difference in PCR product or band size on an agarose gel. Likewise, let's take a look at these other mutations. So in population B, chromosome 2, the distance between the two primers, I'm going to arbitrarily say, is 500 base pairs. So with a 200 base pair deletion in population A, that would produce a 300 base pair product, again, using the exact same primers to amplify two different individuals, one from population A and one from population B, will produce two different PCR sizes. And then if we look at the ancestral allele, chromosome 1 in population B, it's, for example, in this example, 350 base pairs long. With the 100 base pair deletion on chromosome 1 in population F, that means the distance between the primers will be 100 base pairs less, or 250 base pairs long. So then if we look in the F1 individuals, we can see that each of these individuals is heterozygous for one of those deletions or insertions, because each of those individuals inherits one version of chromosome 1 from one parent, the ancestral version from the other parent. We can switch to chromosome 2 and see how one copy of chromosome 2 is inherited from each parent, a wild type version and the mutant version containing, in this case, a deletion. And the same is true for chromosome 3. You get an ancestral version from the parent on the left and a deletion-bearing allele from the parent on the right. So each of the two P0 parents, populations A and B, are homozygous for each of these indels, and their offspring then is necessarily heterozygous for each. And then we cross those two F1s to produce F2 organisms that will have all manner of different combinations of heterozygosity and homozygosity at each different gene and each different chromosome throughout the genome. So this process of creating F2 hybrids creates the genetic diversity among siblings in that generation that lets us start to tell if some genotypes of F2s are less healthy than others, those that show evidence of hybrid dysfunction. So let's take a look at what would happen if we used these PCR primers, which I should mention are numbered according to the chromosome, So we have primers 1F and 1R that amplify this locus on chromosome 1, 2F and 2R, the forward and reverse primers for chromosome 2, and 3F and 3R. So if we used these primers to amplify all of these 4 individuals, let's take a look at what that agarose gel would look like. So for example, let's take a look at population A. This individual, the P0 individual, is shown once for each primer pair. So in the lab, when you're using indel genotyping and gel electrophoresis to identify genetic incompatibilities, you need to take each individual and test it with each different primer pair, in this case 3. So that's why in primer pair 1, we have the P0 individual A and its genotype, individual B, this person, worm, and its genotype, and then one of the 2 F1s is shown. And that's because the 2 F2s, 2 F1s are identical, so there's no reason to genotype them both. So individual A has the deletion. They're homozygous for the deletion. And in this case, we said that the deletion size was about 250 base pairs. So that's that one band on the gel, indicates that both copies of that chromosome, both homologs, have the deletion. They produce the smaller product. If we look at population B, because that deletion doesn't exist in this chromosome, that produces a larger band. And this individual is also homozygous. Both copies of chromosome 1 don't contain the deletion. So both copies, all of the copies of the PCR products from that individual will be larger. And then we see what happens when we get to the F1 heterozygote. They have one short copy, short allele, and they have one long or wild type allele. So when we genotype that F1, we see both the long version, like population B, that parent, and we see the short band, which came from the other copy that contains the deletion. So that's how we use gel electrophoresis to determine the genotypes and the inheritance patterns of alleles from generation to generation. The important thing here is to know that if you see a single band, that typically indicates homozygosity. There are 2 different alleles or chromosomes that contain the same genotype. If it's the small band, it's the deletion-bearing genotype. If it's a large band and a single band, that indicates 2 chromosomes in that individual that have the same genotype, in this case, the longer chromosome. And when we see 2 different bands of sizes that correspond to the parental bands, that indicates heterozygosity. And so we can also see heterozygosity for primer pair 2. There's a larger band in population B, there's the smaller band in population A. And then for primer pair 3 on chromosome 3, the F1 is heterozygous as usual, the smaller deletion-bearing product in population B, so here, and then the larger allele in population A, shown there. So that's how we use PCR genotyping and gel electrophoresis to identify the individual genotypes of, in this case, the parents and their kids. And what we're going to be most interested in, if we're looking for genetic incompatibilities, is looking at the F2 generation and beyond. So the next step is to mate those 2 F1s together to produce that F2 generation. So now I've added the colors back to what the F2 generation will look like. By the way, I should have started by saying, don't panic. This is a big Punnett square, but let's walk through it briefly. Each of the rows and columns contains the genotypes of the 8 different gamete haplotypes, that is sperm or egg genetic contributions, that can be produced by an F1. So an F1 could have all 3 derived alleles, red, yellow, and blue, or it could have red and blue, or red and yellow, or it could have red by itself. It could have yellow and blue together, or blue by itself, or it could have yellow by itself, or none of the 3. So those are, if you look at 3 chromosomes, and each has 2 different alleles, that's 3 to the 2nd power, No, that's 2 to the third power, right? 2 to the third. So you've got a 1 of 2 chance of inheriting a particular allele for each chromosome. So half times a half times a half equals an 8, or 8 different possible haplotypes. And those 8 are produced both in, let's say, those are the 8 different haplotypes that can be produced by F1 sperms. So those are the F1 sperm haplotypes. Those exact same haplotypes are created in the oocytes of the F1. And so what independent assortment, Mendel's second law, tells us is that we can get all possible combinations of those 8 different haplotypes with those 8 different haplotypes that will produce this ratio of genotypes in the F2. So the square itself contains all of the possible F2 genotypes. And it's important to point out that some of these genotypes are identical. So it's not that there are 64 different possible genotypes. For example, this genotype, homozygous red, heterozygous yellow, homozygous blue, is exactly the same as that one. So there is some redundancy in this table. The point here is really to point out that, for example, there's only one genotype like this, homozygous for all 3 derived alleles. There are 2 that look like that. There's only one that's homozygous red and homozygous blue, but has no yellow allele, either heterozygous or homozygous. But there are 2, this one and this one, that again have the same genotype. And if we look at the diagonal from the bottom left to the upper right, every one of those 8 different genotypes is the same, heterozygous at all 3 loci we're looking at. So some of these genotypes are more common than others because, again, this whole Punnett square represents the possible genotypes and their relative frequencies. So only 1 in 64 individuals on average in the F2 generation should have this genotype. But 1 in 8, because that's 8 genotypes out of the 64, so 1/8, 8/64ths of the individuals in the F2 generation would be expected to have this genotype, homozygous or heterozygous rather, for all 3 alleles. Now, one really important point for our purposes today is also to realize that for any chromosome of the 3 that we're looking at, or any chromosome in any organism, there are, in this case, 50% gray alleles and 50% colored alleles, regardless of which of the chromosomes you look at. So, for example, let's take a look inside this genotype. For every 1 red version of chromosome 1, there's a gray version. And for every 1 yellow version of chromosome 2, there's a gray version. And for every 1 blue version of chromosome 3, there's a gray version. And that's true through the whole table. For example, even though there are 2 red and 2 yellow and 2 blue there, which in that individual means they'd have 100% red, not 50%. All of their chromosome 1s are red, so 2 out of 2, 100%. 100% yellow, 100% blue. That genotype, when it exists in the population of F2 offspring, is offset by this one, which has no reds, no yellows, and no blues. So if you bother to take the time to count up all of the different possible combinations of genotypes and count exactly how many times does a red square appear in this table, and how many times does a gray square appear at the exact same spot, you'll see that they're exactly balanced. Half of them are gray and half of them are red. The same is true for yellow on chromosome 2 and for blue on chromosome 3. This is an important point to make because this establishes our expectation. If there's no genetic incompatibility, if there's nothing wrong with any of these different genotypes, then when we look at all of the F2 individuals we genotype, we should expect to find that we'll see exactly for any chromosome, half of the alleles being gray or wild type or the original ancestral allele, and 50% should be the colored version, red on chromosome 1 or yellow on chromosome 2 or blue on chromosome 3. And again, Because scientists are blind to the location and identity of potentially interacting alleles, hybrid dysfunction genes, before we start the project, I'm going to take away the colors again so that we're sort of blind to what's going on in this project. So imagine that we actually set up this cross, two F1 hybrids, produce a population, a huge population in the case of worms, which may be the population that we work with. We get a large number, if in the case of worms, it could be 200 or 300 offspring. With all the resources and time in the world, we would genotype every single one of those offspring to find out if there are some missing genotypes, like that red chromosome 1, blue chromosome 3 interaction that I cartooned the proteins of earlier. If those sorts of genotypes are missing from the F2 generation, that suggests that those allele combination or gene combinations are lethal or otherwise dysfunctional. But we don't know that to start. So in this case, what I've diagrammed is picking 10 random individuals from the population of F2s. So imagine I had a petri dish and I saw a bunch of F2 offspring and I wanted to genotype 10. I picked 10 random worms off of that plate and PCR genotype them for each of these 3 chromosomes to look at their genotypes. So the individuals that are circled then are the ones that I'm suggesting that I blindly picked off the plate. I can't see the colors here that I've been using to diagram, so I have no idea what the genotypes of these worms are yet until I genotype them. So these are the 10. I've used Roman numerals to indicate those 10 individuals to give them labels. And we'll use those to track the worms as we genotype them. So here's an example agarose gel result that is a cartoon, of course, of what might happen when I genotype those 10 different worms at each of the 3 primers for each of the 3 chromosomes. So remember, it's important to pick as many F2 offspring as possible and genotype them at as many loci as possible to get the best genome-wide view of what's going on in terms of potential gene-gene interactions. So from that selection of 10 individuals, again, numbered Roman numerals 1 through 10, I've got the genotypes of each at chromosome 1 near the putative red mutation or locus, chromosome 2 yellow, and chromosome 3 blue. So we've seen this sort of a cartoon of a gel before, but we can analyze it. So in this case, individual 1 at chromosome 1 has a single band of 250 base pairs. So that is homozygous for the small or the deletion-bearing allele. That means that individual has 2 chromosomes that each have this deletion. So that's 2 deletion alleles. And in this case, I've color-coded that section of the gel red because the small version is the derived mutation, the mutation that's specific to that population. So this individual has 2 small alleles. A heterozygote has 1 large, 1 small. So what we're going to be doing is we're going to be counting up after we run this gel. Again, this is looking at the genotype of 10 different F2 individuals on chromosome 1. And we want to know how many wild type alleles there are and how many mutant or deletion mutations alleles there are. So we've got 2 small from individual 1. The heterozygote, individual 2, has 1 wild type, 1 deletion. So that's another small. So I'm going to do a running total. So that's 2, 3. Here's another homozygote. So that's 2 more alleles, 5. Another heterozygote. So that's 6 deletion alleles, 7, 8 for a homozygote, 9, 1 from a heterozygote, and then there's 1 more small allele, or a total of 10. And because there are 10 individuals, and each of them have 2 alleles, that means there's a total of 20 different chromosomes that are genotyped and represented on this gel. 10 of them are the deletion bearing, which means 10 out of 20 of the alleles, or 50%, are the deletion version. And again, that's the red allele. So what I'm going to do on this chart down here is for chromosome 1, we just calculated what the frequency is, or the number in the first place, and then we calculated the frequency, 10 out of 20 or 50%, of mutant alleles, the deletion-bearing or red alleles versus non. So in this case, that's 50%. And then we do the same thing on chromosome 2. So again, the smaller alleles on chromosome 2, the ones that aren't the ones shaded in gray are in each of these panels is the ancestral version. So the non-shaded one is the deletion version. So we can go through and count how many deletion alleles there are. So we've got a homozygote there, that's 2, another homozygote, totaled 4, 6, 8, and then 2 heterozygotes, so that's 9, 1 additional, and then 1 further additional, 10. So that's also 10 out of 20, or 50%. Yep. And the exact same thing as you might expect, because I made up this example. There are 10 small versions or deletion-bearing versions of gene 3 or chromosome 3 out of 20 total. Again, 50%. The reason this is important is because when Mendel's laws are working and segregation of chromosomes is random, independent, this gives us a prediction that when we genotype a large enough sample of individuals, exactly 50% of the alleles should be one parent's, in this case a deletion-bearing allele, and the other 50% should be the ancestral wild-type gray alleles. So that's what we see here. That sets up our expectation. And what we're going to do now is see if in the F2 generation, there is a deviation from expected. If we see frequencies that are much higher or much lower than 50%, that can give us an indication that inheritance laws are being broken. And one of the reasons that that can happen is because of hybrid dysfunction and deleterious genetic interactions. So now let's take a look at an example of what happens when Mendel's laws are broken, when hybrid dysfunction does exist in this hypothetical example. So we'll return to the Punnett square, and I will remind you that in the first set of 12 we picked, that included some individuals in the upper left-hand corner. The distribution of the 10 individuals we chose to genotype was random. But if there is a genetic incompatibility present, and it's in genes near the primers that we're choosing to genotype on the chromosomes we choose to genotype, some of these genotypes won't exist in the collection of, in this example, worms that we might pick off a plate, F2 siblings. And that's because in this example, recall that the homozygous F and homozygous H individuals are dysfunctional. In this case, we could assume that's as extreme as being dead, never even developing, maybe even fertilization. Well, fertilization might happen, fertilization might not happen, depending on which genes and which proteins are involved. If, for example, these indicate sperm-egg recognition proteins. But whatever the process is, the end result is that the 4 genotypes in the upper left, there's actually 3 genotypes in the upper left, because, again, the diagonals are identical. So these are 3 different genotypes comprising a 16th of the entire Punnett square. Those genotypes probably won't exist if there's a genetic incompatibility in some of the chromosomes and regions where the chromosomes were genotyped. So in essence, what that means is if there's a genetic incompatibility and it's present on a plate of worms that we might be looking at, for example, we will never see any of those genotypes. That means that when I pick, or you pick, say 10 random worms or any individual to genotype, they're not going to be in that corner because those individuals with those genotypes aren't alive. They didn't develop. They might not have gotten past embryonic development. So we won't see them. We can't pick them. Those genotypes won't be represented on the set of genotypes that we can collect. So here I'm indicating a new set of 10 random individuals that we might pick off a plate if there is a genetic incompatibility. And now let's take a look at what happens when we genotype these 10 individuals compared to the previous 10 individuals and see what happens to our graph of the alleles in this case. So we can start again with the red chromosome. We have, again, adding heterozygote contributes one small allele. Running total, there's another heterozygote, so that's 2 total. A homozygote has 2 alleles of the same small size, so that's 4 total, 5 total, 6, 7 total. And again, because we're looking at 10 worms, each of which have 2 alleles, that is out of a total possible of 10 times 2 equals 20. So in this case, 7/20ths, or about 35% of the alleles are colored, the red version. So what about yellow? We have 2, 4, 6, 7, 9, 10, 11 yellow. So that's what, 11 out of 20 is 55%. Okay. And then we look at blue. And again, the gray alleles shaded here are the ancestral versions, the ancestral sizes, and the colored versions, the ones on the colored background instead of a gray background, are the derived alleles that are distinct between the two populations that we crossed. So those are the ones, the reason we're focusing on these is because these are the alleles that are going to tell us about combinations of potentially incompatible alleles. So in blue, we've got 1 for a heterozygote, 2 for 2 heterozygotes, 4 for a homozygote, 5, 6, 7. So that's 7 out of 20, which is about, what, 35% again. So what does this graph tell us about the potential for a genetic incompatibility to be present on these chromosomes? Well, again, remember we just described that we have an expectation. If nothing bad or good, for that matter, is going on, then we should see the expected frequency of colored alleles versus gray ancestral alleles to be exactly 50%. We should see half yellow, half gray, or half red, half gray, or half blue, half gray, depending on which of the 3 chromosomes we're looking at. Chromosome 1, chromosome 2, or chromosome 3. So in this case, chromosome 2 has a 55% frequency, which is pretty close to our expectation. But what's happening at the red and the blue regions of chromosomes 1 and 3? They're pretty different in allele frequency from our expected value. So what's happening here is what we've plotted on the y-axis is the frequency or the percent of all alleles at 1 primer pair. So again, this is primer 2, which is 1 specific spot on chromosome 2. And we're asking, of those 20 alleles in those 10 individuals, how many of them are the colored allele? That would be the derived allele, or the ones that might be responsible for a genetic incompatibility. The alleles are mutations that arose independently in the 2 different populations, A and B, as they evolved separately. That when you bring them back together in a hybrid might cause dysfunction. So the takeaway here, the punchline, is that when you see 2 different genes or alleles that are underrepresented among genotypes in the F2 generation, that suggests that the combination of those 2 alleles, homozygous red and homozygous blue, is lacking. There aren't as many, there are a smaller percent of those 2 alleles in the whole population of F2 worms, or 10 individuals in this case. The more you genotype, the better it is. that they're missing red and blue together. This doesn't prove that there's a genetic interaction between chromosome 1 and chromosome 3, but it suggests that it's reasonable to take a future look or a further look at whether or not those two genotypes together are absent from the population. In other words, the goal of looking for genetic incompatibilities by genotyping is to get as many of these different genotypes as possible. In an ideal world, we would sequence the complete genomes of hundreds of offspring. The goal would be to find out if there's any particular combination of genotypes, like homozygous red, homozygous blue, that you can never find in an F2 organism, regardless of how many you look at. And if you can never find a single individual with that combination of homozygous red, homozygous blue, and it's just those genotypes that are missing, none of the other ones, that's a strong indicator that that combination of red and blue is somehow harmful, potentially lethal, to the organism. And the reason that it's important and that I've used a third chromosome, chromosome 2, which had a protein that had nothing to do with the hypothetical interaction between the red and the blue proteins, is to demonstrate that this is the type of result you'd expect for the vast majority of genes, loci, chromosomes. That they will behave perfectly normally according to expectation. And then it's only when you see deviations from the expectation that gives us an idea that we should design further studies to investigate those chromosomes and those particular regions of those chromosomes for mutations that might indeed be causing hybrid dysfunction. And I'll conclude with a final point about the analysis of this graph, because you might be thinking, well, 11 out of 20 is 55%. That's not exactly 50%, which is the expectation. And these frequencies that I'm telling you are involved in some sort of hybrid dysfunction are also not exactly 50%, but neither are they 0%. So how do we know how far from the expectation is far enough to merit further investigation or a conclusion that there is some epistatic interaction, genetic interaction between chromosomes in hybrids? And so there are some statistical tests that do need to be performed to make that sort of conclusion of whether or not these frequencies are statistically significantly different from the expected value. So that's one caveat to keep in mind when you're thinking about using genotyping of a limited number of individuals at a wide or small number of loci to determine whether or not there is hybrid dysfunction occurring. In sum, really quickly, the whole idea here is generate F2 hybrids, genotype as many as you can, and look for missing combinations of alleles using whatever sort of genotyping method here we discussed, PCR for insertion deletion mutations, and then agarose gel electrophoresis. But whatever the technique, the idea is pretty straightforward. We have here the Punnett square that tells us the types of genotypes and their relative frequencies that we should see in a neutral expectation in an F2 generation. And if we find, after genotyping a large and random sample of F2s, that there's some deviation from this prediction that's shown in this Punnett square, that can be evidence of, well, at least it's evidence of non-Mendelian inheritance. And one of the reasons for non-Mendelian inheritance can be genetic incompatibilities that are causing hybrid dysfunction.